Hello friends and welcome to the second part of the genetics lecture. This is Soumyajit and I am here to fix your need for learning genetics. In case you missed the first part, hop on to the I button on the top left or go through it. But in case you're just here for fun, let's do a sneak peek of what we covered in the last video. We had learned what genetics was all about, the study of heredity and variance. We got acquainted with the terms of genes and genomes. And we were introduced to the father of genetics, the super monk Gregor Johann Mendel. I forgot to shout out Reginald Punnett, one of the admirers of Mendel, after whom the Punnett square is named. We learned how to solve Punnett squares ourselves and are definitely very proud of it. We also learned about the monohybrid and dihybrid crosses of Mendel, the laws that govern them. Starting off this session, we will see some instances where the classical Mendelian genetics do not apply. The first case is the classic example of tallness of an individual. What would you expect when two parents of average height have a kid? A very tall guy apparently. But how? This is an example of polygenic inheritance, a number of genes that determine the outcome of a single character. The effect is additive and depends on the dosage of the genes that affect the character. This is also seen in the case of human skin color, and it generally follows a binomial distribution in the population. Next we look into a Snapdragon plant flower. We know that the red allele is dominant over the allele of the white flower. So in the first filial generation, we should get all red. What? A pink flower? But I thought the genes don't mix. This is not an art class, or is it? What actually happened was neither the red or the white allele could completely dominate over each other, and the heterozygous offsprings are therefore pink. This is a classic example of incomplete dominance. From a stroll in the garden of the Snapdragons, we visit the nearest blood bank for learning about the blood types of humans. Just ignore the vampire on the corner sipping a drink, okay? The blood groups of the individuals depend upon the type of proteins and sugars present on the surface of the red blood cells, or RBC. These are the A and B antigens that determine the A and B blood group respectively. They can both be present, making the blood group AB, or both be absent, making the blood group O. Now, there are many other factors that contribute to the blood compatibility of humans. But those topics are for a different day. We observe that the antibodies present in the serum of the individual is complementary to the antigens present on the surface of the RBC. So what happens when an individual of blood genotype A mates with someone with blood genotype B? The kid inherits the blood group AB. But why? This is because the blood group is determined by three genes, one of which does not make any antigen, while the other two makes A and B respectively. If you see the genotypes and phenotypes, IA and IA gives rise to the A blood group, but so does IA and small I. But in case of IA and IB, both the genes are equally dominant. Thus, you get a hybrid of A, B blood group, which accounts for the co-dominance in the individual. This is also an example of multiple alleles contributing to the phenotype. Moving on to something a bit more somber, we discuss about the sickle cell anemia. This disease is caused by a defective hemoglobin that hampers the functioning of the RBCs by assuming a sickle cell and clotting the blood vessels, among multiple other consequences throughout the body. This is a classic example of a pleiotropic gene. If you notice, the first filial generation, the homozygotic recessive diseased individual perishes, but the heterozygous carriers are responsible for passing the disease from one generation to other. The last section of this video that is completely depressing is about lethal genes. I swear it's going to get better from now on. These genes or alleles, as the name suggests, cause the death of the individual that inherit them. These are generally recessive in nature and are normally found on so-called non-essential genes. Now, essential genes are indispensable due to the survival of the organism. Non-essential genes, however, mostly are regulatory genes that are essential for the development of the organism. Thus, most lethal genes originate by mutations in the gene responsible for growth and development. These genes are rarely dominant in humans because the individuals often perish before the transmission of the genes to the next generation. One such example of a dominant lethal gene is the one that causes Huntington's disease. Since the onset of the disease is slow, the allele can be passed on to the offsprings. We just saw a weird looking flowchart when talking about Huntington. What is that? That is called a pedigree, a chart that illustrates the passage of genetic traits from one generation to the other. The females in the pedigree is represented as circles while the males are represented as squares. The horizontal lines indicate the act of mating, except when it is above the shapes, which determines offsprings of the same pair of parents. The shapes shaded are affected individuals, while the healthy individuals are clear or white in nature. The shapes with a slash across represent a deceased or dead individual. So in this case, the dominant allele is propagated from one generation to the other, just because the onset of the disease is late. A familiar Punnett square determines how the gametes are transmitted from parents to the offsprings. But this was for a dominant gene. What happens if the disease-carrying gene is recessive in nature? Comment the answer down below because we already know the answer from one of the previous part of this video. Now, the burning question in our minds: was Mendel wrong? Before you get the pitchforks out, let's discuss why he didn't get any response at the time. He proposed a theory of non-blending inheritance, which from this video itself we have seen there are many exceptions of. For example, the incomplete dominance of the Snapdragon flowers. He proposed a mathematical model which was frowned upon by the biologist at the time. And then the main problem was that he could not provide any physical evidence of the factors he spoke of, because we did not know of genes and chromosomes at the time. To his credit, he himself acknowledged to this point, where he noted, with some of the most striking characters, those for instance which relate to the form and size of the leaves, the pubescence of the several parts, etc., the intermediate indeed is nearly always to be seen. He spoke of incomplete dominance himself. So, how did the story unfold? Let's learn about autosomes and sex chromosomes first. Genes are generally located on chromosomes, and these chromosomes almost always come in a pair. Autosomes are the chromosomes harboring most of the genes required for cell homeostasis, while the sex chromosomes determine the biological gender of an organism. The cells having both the sets of chromosomes are called a diploid cell, whereas a cell having a single set of chromosomes is called a haploid or a gamete cell. In case of humans, the Y chromosome lacks most of the genes of the counterpart X chromosome. So in such cases, the genes in the X chromosome are always expressed, or it dominates over the phenotype that are present on the X chromosome. Now we get to know about Thomas Hunter Morgan, a Nobel laureate who worked on fruit flies, checking which flies have red colored eyes and how it is propagated. In case of the female flies, the dominant red allele will have red eyes, whereas the males who inherit the recessive white allele will have white eyes because the Y chromosome does not have an allele for the gene. This provided the first strong evidence to the chromosomal theory of inheritance, proposed by Walter Sutton and Theodore Boveri. Based on the inheritance pattern of the eye color, Morgan concluded the eye color must be located on the X chromosome. So let's compare the theory of Mendel and the chromosomal theory of inheritance. We know that the chromosomes come in pairs, what we now call as homologous chromosomes. Mendel also mentioned his factors come in pairs. We know that one chromosome comes from each parent. This was also the case of the Mendel's laws of inheritance. The chromosomal theory of inheritance says that each chromosome comes from each gamete. This was also the case in the law of segregation by Mendel. One chromosome comes from each gamete and are independently assorted, which is eerily similar to the law of independent assortment by Mendel. Thus, can we say that the gene is the unit of heredity, and the chromosomal theory of inheritance therefore supports the laws of Mendel? In summary, we can comprehend that we have learned about different phenomena and diseases that do not obey the Mendelian laws. We thereby questioned if the Mendelian laws were even valid. We learned about the chromosomal theory of inheritance and thereby learned a valuable lesson. The discovery of science is progressive, and although Mendel could not figure out the whole picture due to limitations of his time, he paved the way for future scientists to investigate genetics. Thank you for watching. Show your love by clicking the like button. Consider a subscribe so we can bring you more of these videos, and see you next time.